This is a very special t-shirt. As you can see, this is an evolution phylogenetic tree. And this is a lab t-shirt actually from my lab when I was at UC Davis. So it was kind of cool because bicycle is the symbol for UC Davis. So just kind of cute. Okay, we can get started. And today and the following week, we are going to talk about the methodology and the principles for phylogenetic analysis, i.e. the inferring the phylogenetic relationship using different methods. Today, I'm going to give you some of the concept, general introduction, and I'll talk about the very simple, the simplest way to reconstruct the phylogeny, the distance methods, and also the parsimony methods. And next week, I'm going to talk a little bit more about the Measurement likelihood and Bremer inferences, and also some other methods which is less used, and also how to evaluate the trees. And but today I'm going to talk about that as well. So let's begin with some of the introductions. And the methodology I'm to talk about will be based on two different criteria. One is called the distance method, and the other one will be called the discrete data method, and which means the character-based method. So many of the phylogenetic analysis can be categorized based on the two different approach, and I will talk about that in detail on distance and discrete data. And for the distant data, including the UPGMA, neighbor joining, and minimum and evolution, those are pretty commonly used before, and neighbor joining is most commonly used nowadays for distant method. And today I'm going to talk about the parsimony in particular. The other two will be left out for next week. And before I really started that, this is just some of the introductory slides I've been using it for journal purpose for evolution or molecular evolutionary lecture. And just give you some idea about the how this phylogenetic analysis concept was evolved over time. And the basic idea for the molecular data itself, of course, is the discovery of the DNA structure and so on. But the characteristics or phylogenetic methods occurred way before that. It occurs about 1930s, and actually 1920s. There's some discussion already. So that's way before the discovery of the DNA and also the nucleotide sequences and so on. So what did they use? And basically are those morphological data. So the phylogenetic analysis method was developed in the beginning for the purpose using the morphological data. But the same method can be applied to DNA or amino acid sequences as well. And in fact, amino acid sequences was first applied to the phylogenetic analysis before the nucleotide sequences, because it was, but the timing was very similar, about 1950s and 1960s. And several different models and approaches has been used, and like by Sanger or Alan Wilson, and also some others. But the actual breakthrough happens only after 1980s, and that's after the PCR and the sequencing method was widely adapted and become cheaper and cheaper, so everybody can have a large amount of data. And the methodology also improved owing to the computer speed as well during the 1990s. And the methodology like the measurement likelihood and are largely adapted from 1970s. That's by Feldsenstein, Joe Feldsenstein at that time. And before that, the parsimony method was largely developed earlier than 1950s. So we'll keep that in mind. So there's the method I'm going to talk about are already almost 1 century, well, not 1 century, maybe 80 years ago. So it's, in a way, it's kind of mature. It's has been discussed a lot. Even that, there are still quite a lot of methodological concerns about how to really use that, and there's some problems associated to it, and we're talking about that in a minute. And nowadays, there's another problem is that we are having more and more data on genomics and genomic scale. So, which also poses another problem on the phylogenetic methodology. But we'll worry about that in a later lecture. And the three major contributions in my mind for the molecular evolution is these three guys. And the first is Willy Hennig. It's who actually contribute a lot on the characteristic, and which means the original phylogenetic methods. This is one of the founders for that. There are several others, of course. And for the model and the neutral theories for molecular evolution was developed by Muto Kimura, and talking about the mechanisms itself on the molecular evolution. But in my mind, Joe Felsenstam is the lead guy to really combine the models and also the methods, to apply to phylogenetic analysis. He is the one who really used the maximum likelihood methods on phylogenetic analysis. So, and he is also the author for this book, Inferring Phylogenies. And this is a very, very nice book. I think I already talked about it in the very beginning. And this book, and also the other one, which is newer, the Phylogenetic Handbook, edited by Philip Lemay. And these two are the best references for the two lectures I'm going to give this week and next week. So for anyone who really want to know more about the phylogenetic analysis, this is the reference you should go to. And there are probably 10 more books you can read, but these two are the best. And but only if you are really serious about that. Otherwise, it's probably too difficult for you. You can find some other simple ones.